Today's lecture is today's lecture is gene expression in eukaryotes, or gene expression 2. This is very much a continuation of the previous lecture, so please make sure that you watch the previous lecture before you watch this one. Today what I'd like to talk about in detail is regulation of or control of gene expression at the level of DNA, and then move on to the level of RNA and the level of proteins. So eukaryotic regulation of gene expression is a little more complex than that of prokaryotic regulation that we discussed before. There's many reasons that eukaryotic organisms might change their levels of gene expression. Could be response to light in terms of plants, right, to determine when flowering happens each season. Could be response to hormones or other regulatory molecules. Could be developmentally regulated, for example, when an organism goes through puberty is a classic example. And there's also specialized cells. So different cells express different genes at different times, and some barely express certain genes at all. And that makes sense because remember, every cell in your body has the exact same DNA sequence, but they have very different functions. So they have to have different levels of gene expression to achieve that end. Okay, so let's first talk about a little more, a continuation of last time, a little bit more about expression of genes at the DNA level, so controlling expression at that level. Okay, so transcription is controlled by many factors. There's transcription factors that control gene expression, and they have different domains that are very important we got to remember. So things we've talked about before, but just want to make sure we highlight them here. So there's DNA binding domains, there's activation domains, repression domains, and then domains that allow proteins to interact with each other. Again, things you've seen before in this course, but I just want to emphasize them because they're very related to the topic at hand today. Okay, so whenever you have eukaryotic promoters, they might have multiple regulatory elements. So in other words, here you see 3 different eukaryotic promoters. You'll notice each one has a core promoter, right? So that has to be there. I think of that as being essential. If you don't have a core promoter, you're not getting transcription at all. But then they also have a regulatory promoter that's a little upstream of the core promoter. And what I would do is I would think of the regulatory promoter as sort of like a fine-tuning of transcription. In other words, the core promoter says, does transcription occur, yes or no? The regulatory promoter says, to what degree would transcription occur? The other thing I want you to note in this slide is that you'll see consensus sequences all over the place. So just familiarize yourself again with what a consensus sequence is. TA, you'll notice these other ones here, OCT, right? These are just, obviously there's no O in DNA, right? But these are just labels for different consensus sequences that you see. Probably a better one here is CAAT, just different consensus sequences that you see. Okay, when we have transcription in eukaryotes, it's controlled by multiple DNA elements. We just on the last slide talked about the core promoter, about the regulatory promoter. But there's some other players that we have to discuss here. So for transcription to occur at all in eukaryotes, this is the basic set of proteins that you need. And the basic set is called the basal transcription apparatus. And what that is composed of is the RNA polymerase itself, which does the transcription. It's a huge enzyme. I think of like a sumo wrestler walking a tightrope. And you can't have that sumo wrestler walking that tightrope unless some people are helping to hold him on, right? And so you could see that here these transcription factors are also part of the basal transcription apparatus, and they help recruit the RNA polymerase to this site, to this promoter site. So transcription can happen right after the promoter, going to the right here. So the basal transcription apparatus is composed of those proteins, and that is required for transcription. You have to have that for transcription in eukaryotes. There's some other players that are optional, that enhance transcription or increase it, but are not necessary. Things called mediators, co-activators, other things that are called transcriptional activator proteins, like this guy and this guy over here. And they can all enhance transcription in 2 ways. And so one way is by physically tethering themselves to the RNA polymerase to recruit it to that promoter site. And that's called scaffolding. So the first way I would write here is scaffolding, just that physical touching of the proteins to the polymerase. The second way, though, so number 2, is by their acetyltransferase activity up here. And what that does is, remember, when you acetylate DNA sequences, you open them up and allow them to be transcribed more easily. So that's the second way. There's also DNA sequences that could further increase transcription. So the previous things I mentioned were proteins. But here you could see that we have a DNA sequence called the enhancer. And what the enhancer does is the enhancer is a DNA sequence that recruits proteins called transcriptional activator proteins, the same thing as before. And what that does is you actually get this like upper level structure or folding of DNA, and the enhancer will bind to the RNA polymerase. Again, the scaffolding through the transcriptional activator protein and the mediator. Just think of it as another way, this wrapping of the DNA, another way to help stabilize that RNA complex. The thing I want you to note about the enhancer is not location dependent. So the promoter, the core promoter, and the regulatory promoter have to be just upstream of the start site of transcription. The enhancer can be located quite a distance upstream, but it comes in close proximity through this folding of the DNA. So the enhancer is not location dependent. Okay, there's other players, again, that you have to keep in mind. So things that are called insulators. And what insulators do is they basically muffle the effect of an enhancer. That's all they do. Or there's things called silencers that are the opposite effect of an enhancer that decrease transcription. So I think of this, I think of as enhancers and silencers trying to either increase or decrease transcription. And what insulators do is their DNA sequences, and things called insulator binding proteins bind to them, but their DNA sequences that basically muffle the effect of either the enhancer or the silencer, so bringing things to a happy median. So in other words, this enhancer number 2 here can affect this promoter, right, and hence gene B, but since the insulator is between enhancer 2 and this promoter over here, enhancer 2 cannot affect this promoter and cannot affect the transcription of gene A, and then vice versa for this other enhancer over here. So that's the job of the insulator. There's also compaction of DNA issues that we could talk about here. So the degree to which chromatin is compacted, in other words, whether it's condensed or opened up, can affect transcription as well. And we've alluded to this before. This is really the field of epigenetics. And it's an extension of a theory proposed by a scientist about 200 years ago in the early 1800s. His name was Lamarck. We'll talk about his theory in a second. Now, just to be clear, the way he proposed his theory was completely wrong. Okay, so we don't want to say what he said was right. But if we broaden his theory, we can actually sort of form the modern theory of our field of epigenetics, which we'll talk about in a second. But there's 2 things I want to focus on in this slide. So one of them is these GC islands. There's all these GC islands that can be methylated in DNA, and what that can do is that can actually decrease transcription. There's other things called histone proteins, which we've talked about before in this course. So here you see a nucleosome, here you see a nucleosome. And what happens is whether the tails of the nucleosome, so the histones within the nucleosome, those tails, if they're normally sort of positively charged, what happens is they hug the DNA or they squeeze the DNA and they really condense the DNA that's wrapped around them. However, if you're able to acetylate these tails, what happens is you relieve that positive charge, they open up, and you can see the DNA looses around that nucleosome structure, and then you get enhanced transcription. Methylation has the opposite effect. It actually decreases transcription because it causes those tails to further squeeze the DNA that's wrapped around them. So it's a very interesting field here. So here's a little example of this epigenetics in action. And what I'd like you to do here is draw a vertical line right here on your slide. Because really everything to the left of this line, think of it as happening in the winter. And then everything to the right of this line here, think of it as happening in the summer. It's the same picture you're looking at on the left and the right. It's just that it's different seasons. Okay, so think of it that way. Okay, so what happens? So here we have a gene called flowering locus C. When the DNA is acetylated, so when it's opened up, flowering locus C can express. We get transcription, we get translation, and we get a translational activator protein that actually leads to the repression of flowering. Right? So we don't get flowers when flowering locus C is activated. On the other hand, if we have the effect of another gene here, so flowering locus D, now this is in the summertime now, I'm going to talk about in a second. So to the right here, we're in the summer, or in the spring, if you will. When we have flowering locus D, so this is a new gene we're introducing in the problem. Here's C, the original one, right? But here's a new one, D. When we get transcription of that gene and translation, it produces an enzyme called a deacetylase enzyme. And this deacetylase enzyme basically will deacetylate the flowering locus C gene. And what happens then if you're deacetylating, it's the opposite effect, than if you were to acetylate. So what happens here is it restores that chromatin structure. It condenses flowering locus C. So now we don't get transcription, we don't get translation, and we cannot repress flowering. And so now we actually get flowers. And you can imagine that flowering locus D, the stimulus that activates that, is actually changes in temperature and changes in light. As we get warmer temperatures and more we get lengthier days in the springtime. Okay, so let's relate all this to Lamarck and to Darwin a little bit. Let's put all this in evolutionary context. So in the early 1800s, I believe the year was like 1809 or so about 50 years before Darwin published his theory of evolution, a scientist by the name of Lamarck published his theory of the mechanism of evolution. And what he said was this, and so you can see this in generation 1, generation 2, generation 3, this giraffe, right? He said this giraffe's necks will get longer over time, okay, so change over time, there's the evolution. But he said the theory was use and disuse. Now I'm going to say his theory, but keep in mind, his theory is the way he said it is completely wrong, okay, so this is not something that we hold true today. But I just want to express his theory so you could see what it was. So it was use and disuse. He said the giraffe kept reaching higher to get more foliage, more food, and so it kept, basically, straining its muscles in its neck, straining its muscles in its neck. And he said what happened was because of that, the offspring had longer necks, right? Because it was used. If the giraffes did not use their necks, the opposite would happen. They'd probably shorten over time, right? But he said they used them, so it got longer. This individual reached for more and more food, it got longer, right? Until the giraffes could access all the food that was at their disposal. So now he said, what's the cause of this? And this is where his theory really goes awry. The cause, he said, was there's some vital force or some electricity in the neck. He didn't really define it on us. It was very abstract. But some electricity or vital force that, enabled the neck to lengthen over time. So now that's something we don't hold true today. But if you broaden his theory, and we'll talk about it more in a second, so if you broaden his theory and say what happens to you in your lifetime can affect your offspring, his theory does have some validity in an epigenetic sense. Okay, so we're going to talk about that a little bit in today's lecture. Okay, let's compare and contrast that to Darwin's theory. Well, Darwin's theory, go back to Lamarck a second, notice he had one giraffe per generation. Keep that in mind. Okay, with Darwin's theory, you cannot illustrate it with one giraffe per generation. You have to have at least a few, right? At least 2 or 3. 2 would be the minimum, I guess. But at least a few giraffes to illustrate his point. So he proposed a different mechanism of evolution, and this is in the year 1859 when he actually published his work. So 50 years after Lamarck. So by this point, everyone thought Lamarck's theory, did not hold water. But what they did do was they said, okay, well, Darwin proposed his theory. And what Darwin showed was he said that for evolution to happen, there has to be variation. There has to be differences in the lengths of the giraffes' necks to start with. That's a prerequisite. And then he said what happens is, it's not what happens during their lifetime, it's just that the giraffes that happen to have longer necks will happen to be able to reach more foliage, more food source, and so they'll be healthier, they'll be more fit, and they'll be able to produce more offspring in the next generation. That's what he said. And basically, if they can produce more offspring, their average child, right, will have the same neck length that they had, and they'll have some that have shorter necks, some that have longer necks. But if only the longest neck giraffe is reproducing the most in this generation, the average shifts, right? So the average is now like them as opposed to what the average used to be, and that persists through the generations. He called this natural selection or survival of the fittest. Keep in mind that the word fit in an evolutionary context is sort of circular logic in a sense, which is okay. But fit doesn't mean what you look like or your speed or how healthy you look. But what fitness literally means is who has the most offspring. And those offspring have to be viable. They have to be able to have offspring. So that's what fitness means. So again, you don't know who's most fit until you see how many offspring they have. But that was Darwin's theory. So you might say, okay, well, who do we believe? We go with Lamarck, we go with Darwin. Well, we can't go with Lamarck as he said it, right? Darwin's theory, as he proposed it, 150 years ago, actually is still very valid, and really everything he said is pretty much right on the money. But you have to keep in mind that we've added to his theory over the last 150 years. So his theory is not just the say-all and end-all of evolutionary biology, right? Certainly is the bedrock. I mean, it is the foundation of evolutionary biology, but we've built upon it. And so now people say, okay, we have Darwinian evolution, but we also have Lamarckian evolution. Not the way he said it, right? So Lamarck was wrong. But if we brought it and say what happens to you during your lifetime can affect your offspring, then it does hold some water. And what we mean by that is if you have epigenetic changes that happen and they're passed through the generations, that's a form of Lamarckian evolution. So just to be clear, differences not in the genes that are passed on, right, with Lamarck, the versions of the genes are the same, but it's the expression patterns. What genes are expressed at what time? That's Lamarckian evolution. So he did not say it that way, but if we give him, let's throw him a bone. If we broaden his theory, that's what modern day epigenetics is. So modern day evolutionary theory really sort of combines ideas from these two individuals, as well as many, many other things we haven't even mentioned in this lecture. Okay, so let's talk about control of gene expression at the RNA level, transcription, processing, those things. So we're looking at the middle segment of our central dogma here. Okay, so again, just a reminder of the central dogma, right? DNA transcribed to RNA, translated to proteins, different in prokaryotics and eukaryotic organisms in the sense of the spatial temporal separation in eukaryotes. Okay, so just a little review, so don't forget that. Okay, let's look at this. So what could happen is, as we mentioned before in previous lectures, if we have abnormal splicing that happens during or after transcription when the RNA is processed, we can have diseases that result like this, something like progeria, right? Remember, progeria is basically, a disease that happens. We have abnormal splicing. We talked about it before, right? But the progerian protein is shorter than normal because of that abnormal splicing, even though it's a silent mutation, right? But the abnormal splicing causes these issues. So we end up having a protein that's shorter than normal, and it has this farnesyl group right here. This farnesyl group recruits things to the nuclear membrane and causes those defects you saw on the previous slide. Modern therapeutics are trying to come up with drugs that are farnesyl inhibitors that get rid of the farnesyl group. You can't really give these individuals normal lamin A protein, because they already produce some normal lamin A, right? Only one of their genes, only one of their lamin A genes are defective. But you can't give them normal lamin A to fix the problem. Really, you have to get rid of the farnesyl group. And the reason is because you can see this guy here. Imagine another lamin A protein right next to it that doesn't have the farnesyl group. What happens, though, is lamin A forms dimers. So even if half your lamin A is normal, if the other half is not normal and has this farnesyl group, they're going to form dimers, and they're both going to be taken to the nuclear membrane. So it's almost like the adage, one bad apple spoils the bunch, that type of thing. So it's a very interesting, connection to control of gene expression. Now there's also things called regulatory RNAs, and these regulatory RNAs basically regulate gene expression by 4 mechanisms here. I'm just going to talk about 3 of the 4, but these are 4 mechanisms that happen. So I just want to show you a diagram. This is a nice connection to lab, so something we've done in lab at this stage. Remember, when double-stranded RNA is injected or introduced into an organism, an enzyme dicer comes, chops it up into siRNAs. Those small interfering RNAs guide a complex of proteins called the RISC complex to the endogenous messenger RNA. Again, this is review, right? Things we talked about in lab. The slicer activity of the RISC complex chops up the endogenous RNA. We get degradation, and that's how we control gene expression. So the organism is transcribing their DNA to RNA, but what's happening is you're destroying the RNA, so it doesn't really matter. The same thing can happen in a slightly different manner. So the whole beginning part here is the same. But notice here that we have things we call them microRNAs instead of siRNAs. We call them microRNAs because these microRNAs don't hybridize perfectly to the endogenous RNA. There's a little mismatch. That's what this bulge means here, a little mismatch. If we have a mismatch, the slicer activity of the RISC no longer is activated. So the RISC complex, instead of cleaving the endogenous RNA, just sits on it. If it's just sitting on it, you could see that if it's just sitting there, translation is not going to happen, and so we still get control of gene expression, but just through a slightly different mechanism. We're not destroying the endogenous RNA, but rather what we're doing is just inhibiting translation. There's another way, too, that regulatory RNAs act, and I have this on a different slide from the previous two, because here really the action of the siRNAs, or the small interfering RNAs, is at the DNA level. So it's going back to the DNA level as opposed to destroying RNA or inhibiting translation. So here you have these siRNAs that are produced from the same process as mentioned on the previous slide. But what happens here is they actually base pair with the DNA itself, right? And what happens is they recruit something called the RITS complex. RITS just stands for, over here you can see, RNA transcriptional silencing. And the RITS complex recruits methylating enzymes. And you probably know the story from here. When we methylate DNA, we condense it. When we condense it, we inhibit transcription. Right? So another way that regulatory RNAs can come into play. The final thing I want to touch upon, just in a general sense, to give you a taste of it, is how we control gene expression at the level of proteins or at the level of translation. Okay, so let's focus on that. And so you could see here that what happens, and we're going to cover this very generally because we covered the other things in a bit more depth. But here, to give you an idea, right, we could have T cells when they're exposed to an antigen like a virus. What happens is they increase the availability of things called initiation factors, which we discussed in our lecture on translation. Initiation factors, when we increase them, they're going to increase the availability of ribosomal subunits. We're going to get increased translation to occur. This increased translation, so we're over here now, increased translation is going to activate the production of more T cells. So we have one T cell in the beginning, but when we're exposed to an antigen, we're basically increasing production of T cells. So now we have 4 at the end. So just a general way to show you how we could affect control of gene expression at the level of translation. So in the previous lecture and in this lecture, we talked about how gene expression can be controlled at all these different levels. Now keep in mind, you might say, well, why not control it at the beginning, at the level of DNA? That seems most efficient. I would agree, right? But there's 2 things. No one said nature had to be efficient, right? So that's one thing. It didn't evolve that way. But the other thing is, in a biotech sense, there's benefits to be able to control gene expression at this level, or this level. If you destroy the DNA sequence, the gene, or you permanently silence it, sometimes you can't recover from that, right? That might not be a goal you want. You might want to regulate gene expression for a certain period of time, but then unregulated at a different period of time. So a target in that sense of destroying the RNA, which is just sort of a temporary solution, if you will, might be a good thing to take advantage of. So I'm not saying nature operates with this intent, but if we hijack this system and we use it in a biotech sense, then that's how we can take advantage of it.